Welcome everyone to the workshop on translatability and regulatory affairs for EURONANOMED. EURONANOMED is Iranet co-fund. It's a project funded by Horizon 2020. The main activities or main goal is to funding collaborative research and research and innovation on nanomedicine. But there are other activities that are as important as that, as we have the opportunity to have now, is a workshop, a training workshop on translatability of results that I hope that will be very fruitful. So I stop here and I will give the screen to Olaf Rothaus, who is the Network Steering Committee Chairperson. So I hope that you enjoy with the workshop and you have nice discussions. Olaf, the floor is yours. Thank you so much.
Okay, thank you very much, Ignacio, and welcome everyone to our workshop on regulatory affairs. It's the fourth workshop that we are doing in this way. I just want to give a very short introduction and I try to show you my screen. Okay, I think it worked. Well, it's a workshop on translatability and regulatory affairs. As you know, the aim of EURONANOMED, and we are now in the third EURONANOMED, so we started in 2009 and we are funding nanomedicine projects since 2009. The aim of EURONANOMED is to develop innovative diagnostic and therapeutic solutions to solve unmet medical needs and to have transnational and translational projects. So of course, we want to make people from different European countries working together, but we also want people to make people work together from academia, from clinic and from industry. So that's the translational aspect. And of course, we want to make patients benefit from nanomedicine. And we have seen in the very beginning when we started talking about nanomedicine and we started with EURONANOMED that there are some challenges to get drugs, especially nanomedicine drugs, to the market. On the one hand, there are regulatory issues. You have to think about safety and efficacy studies of clinical trials and things like that. You have to think about preclinical manufacturing, GMP. And of course, you have to think about IP issues. You have to look, do you have freedom to operate? Do you have an IP strategy for your products? And well, there are many challenges that make that a lot of interesting ideas from the lab do not reach the market and do not reach the patient. And so we say that, or we noticed that those translational issues have to be taken into account already at an early stage, already at the research stage of the projects. And so it is important to have knowledge, at least basic knowledge about translational issues when you are doing such a project. And that was the idea to have from time to time in EURONANOMED for the projects who are funded in EURONANOMED, these kind of workshops. So to provide to you information about regulatory issues, of course, to show you some interesting and encouraging examples of people, of young companies who made it to the market with nanomedicine product, and to introduce to you some important European initiatives in the field of nanomedicine, where you can go, where you can get information, where you can get also advice, and where you can work with. So we want to help you to raise the success and the translatability of your funded EURONANOMED project. And this is what we will do today. So we have 6 talks during this workshop. We will talk, or you will have a talk about the general requirements of approval of nanomedical products. You will have a talk about medical device regulation. Then we have 2 interesting projects that we want to introduce to you that made it to the market and made it to the patient, Ruby Nanomed and Live Nano. Then you will hear something about intellectual property rights management. And finally, you will be introduced to some important and interesting European initiatives in the field of nanomedicine. That's the program from today. And after each presentation, you will have the possibility to ask questions. There are two ways to ask your questions. On the one hand, you are watching us on YouTube. You have the chat window on YouTube, so you can type your questions in the YouTube chat, and the questions will be read to the presentators, and they will be answered. To ask a question in YouTube, you will have to be registered on YouTube. If you are not, you have a second possibility, and I put an email address here, enm3@iscii.es, and this is the email address where you can send your questions, and then also they will be answered after the presentation. It depends a little bit, of course, of the time that we have. So I will stop now this short introduction. So the next speaker is Basil Acra. He started his scientific career at the University of Munich, working on tissue engineering. And then he went to the TÜV Süd, which is the biggest notified body in Europe. And he gave advice on regular approval of medical devices, combination devices, and ATMP. And he was vice president for global strategic business development. And 2 years ago, in 2000, or 1 year ago in 2000, he founded his own company, a consultancy company, which is called QNIQ. And well, he will give us interesting information about medical device regulation. Please go ahead.
Thank you, Olaf, for the introduction. Thank you for inviting me here to speak about the medical device regulation, and especially with the focus on nanomaterials, giving you some insight about how the requirement of the medical device regulation going to influence the CE certification of medical devices, including nanomaterials. I will share my screen, and I hope that you can see it now. And now I'm moving on. Perfect. So I think you see my screen, and Olaf just started with an introduction. So last year, in June 2020, I decided to in a company on consultancy. Beforehand, when I was working for notified bodies, I was not allowed to advise and explain the requirement. I was an independent member of let's say, a body in Europe who is responsible for certification of devices, and based on that, advice and recommendation were not possible. So now talking about the medical device and trying to explain to you some of the requirements. Before I start my presentation, I always start with a short disclaimer based on the fact that Europe is undergoing a big change in the legislative framework. We are moving from directives to regulation. And at this moment, during the implementation phase of the new regulation, which I'm going to explain to you in some minutes, we are seeing a lot of interpretation, a lot of new guidance. And based on that, whatever you decide to say today, it's not anymore valid probably tomorrow, because a new interpretation is coming in. So when we talk about medicine, and I think René was explaining to us very clearly about the established requirements for medicinal products, and the ICH guidance documents and the additional requirement on international level, which were already established, and the challenges also with the pharma requirements. When we look to medical devices, they were regulated in Europe for many years, and now we are facing a big change towards the regulation, moving from directive to regulation. And anytime when you implement a change, you know it, even if you are not dealing with regulation, this requires a lot of effort, a lot of let's say, knowledge to ensure that we are addressing all the requirements. And therefore, everything is based on actually current interpretation, and things could change regularly. Now, talking about medical devices, I think COVID showed and made all of us more experienced. If you haven't been working in the regulatory field in the past, you recognize now, based on the COVID situation that every country is facing, that you started establishing some knowledge on how devices, how pharmaceutical are getting to the market. And one identification that a lot of people started to do is understanding what is CE. A lot of people before, they were not really focusing on this, but nowadays, if you go to a grocery mark and you're trying to get a facial mask, the first thing that you do, you look, is it CE mark? You try to understand what actually the approval of that device or that product to ensure that you are getting, let's say, better evaluated devices than getting devices which are not actually checked. And CE marking stands actually for Conformité Européenne, which means that this is in compliance with the applicable requirement in Europe. Now we have CE marking with number and CE marking without numbers. CE marking with number means that the product has been assessed independently by an organization which is named notified body, for example, who did an independent assessment of compliance of that product. And this is why the number is reflecting the digits which are related to a specific notified body who was involved in that process. The whole CE marking idea was created actually with the new approach towards having a better free of movement of goods across the European countries. Nowadays, we hear much more about that based on the Brexit that happened in the UK, the impact on the access to the UK market, but also the access of the European market to UK products, because the basis which was associated with CE marking is now broken down. Additionally, we see that a lot of discussion with regard to Turkey, to Switzerland. So a lot of these things will be coming to us and we will see them in the upcoming years. So the most important thing at the beginning is to know CE marking is important towards having product moving across the European countries where you have an acceptance that those products are fulfilling the European requirements. For medical devices in the past, so in the past I'm referring to actually from the 90s, we got in the 90s directives which are dealing with active implantable medical devices. So are like pacemakers, similar to pacemakers, for example, or directive which is dealing with medical devices in general, such as devices like hip implants, like tubes, cannulas, whatever. They were covered by the directive, which is for medical device directive, MDD. And then we had a directive for in vitro diagnostics. Those directives were like for nearly 30 years untouched. They were amended a bit to reflect the current knowledge, current state of the art. And they needed a revision, a holistic revision, which was actually accelerated based on the scandal that we saw in Europe. So if you remember, we had the PIP, which is the breast implant scandal. Then we had the metal on metal scandal, which led to a decision to accelerate the process of getting regulation published in Europe, which are towards getting higher safety level and ensuring that the product on the European market are performing as intended. Now, talking about the European market, a lot of people believe that the system is very easy and nicely structured. I think René was explaining about BfArM and the responsibilities of BfArMs. But in Europe, we are not like in the US, where we have the US FDA taking care of everything. We have a very complex structure because when we deal with medical devices, the medical devices require the involvement of notified bodies, and you have devices which are based on self-declaration, where a manufacturer declares conformity without a notified body involvement. Those are the devices which are just CE mark without a number. When you have a CE mark with a number, this means that the notified body was involved, and this is actually a risk mitigation step. And this means that more risky devices require notified body involvement. Devices which are just CE marked, those are very low risk devices where an independent assessment of a third party is not deemed necessary. The self-declared devices are nevertheless still under surveillance activity. The surveillance is taken by the member states where those devices are located. So in Germany, Germany has below BfArM, there are also other member states which below BfArM, not member states. We have states like Bavaria, like other places which are taking also surveillance activities and controlling the class 1 devices. Other European countries, they have the same. When we have a device which is under the surveillance of a notified body, the notified body takes the surveillance. But then the notified bodies are by themselves monitored by authorities such as the ZLG, which is actually the German accreditation body, which is cooperating with BfArM, but not underlying under BfArM, because all of them, they are like authorities acting in the system. So you see a lot of complexity. And if you have a device which includes a drug then notified bodies need to cooperate with an authority who's going to be taking care of the drug part. And this authority doesn't need to have to be in the same location where where the notified body is located, but it could be in a different member state. So it's a lot of complexity. It's just a short illustrative demonstration with regard to Germany. And I think if we do that on a European level, then you will get really gray hair because you will not understand what is behind it. It's just important to understand it's not one single stop where you have one organization taking care of everything. The activities are delegated to multiple responsible organizations who can be authorities or notified bodies. And notified bodies are not an authority. Notified bodies are independent third-party organizations who were qualified to be actually allowed to act in the name of the European Commission with regard to certification of devices. And this is why they are actually regulated and monitored continuously by auditing, let's say, by getting them audited by authorities who can confirm regularly that they are still qualified and fulfilling the requirement. Taking this into consideration and considering also the fact that we are undergoing a big change in the European system, we recognize that actually for this change, you need to have qualified notified bodies again. So I told you we were in the past under the directive. In 2017, finally, after 5 years of let's say, finalization of the regulations for medical devices and in vitro diagnostic products, the European legislators, which means Commission, Council, and Parliament, agreed on a final version of the regulations. So they published in 2017 in May the medical device regulation with a transition period of 3 years, and the in vitro diagnostic regulation was a transition period of 5 years. And they clarified that in the upcoming years, then how long devices can be still placed on the market based on the current valid certificate. So this means that every device which is already on the European market today and which was approved will have a deadline where this device will need to undergo a completely new certification to be able to ensure market continuity. And this is a very critical problem because it is actually saying not just that manufacturers need to be requalified, but also the notified bodies which we have today under the current system need to get a requalification to be able to conduct assessment of devices or in vitro diagnostic devices under the new legislation. Now, looking to the medical device regulation, which is the focus today of the workshop, what we are discussing, talking also about nanomaterials later on, we're going to recognize in 2012, when the whole scandals and discussion were happening, we had more than 80 notified bodies with a wide scope enabled to do independent assessment and certification of devices. The scandal happened, and when the scandal happened, then the European Commission started with risk mitigation measures, and one of them was the implementation of the so-called joint assessment, where not just, for example, Germany audited German notified bodies, but multiple member states. So a combination of a German authority, for example, with an English authority and French authority, go and audit the notified bodies under the surveillance of the European Commission to ensure harmonization. This activity led to a reduction of the number of notified bodies from more than 80 to less than 60 after a period of 3-4 years. Now, for the medical device regulation, as I mentioned, every notified body needs to apply again to be able to be qualified, to be able to act under the new legislation. And out of these nearly 55, 56 notified bodies, 50 applied. And you remember I told you this regulation was published in 2017, and 6 months after publication, notified bodies were allowed to start applying. But if you apply, you need to be ready, which means you need to have a system in place which enable you to conduct assessment under the medical device regulation. And in that application, you need to show not just that you have a quality management system which is allowing you to be compliant, but also having personnel which are qualified to conduct the assessment under the new legislation. And all what we have till now are 20 notified bodies who are designated for the medical device regulation. And this is actually also related to the fact that they need to show qualification and competence in every device field where they apply. I told you at the very beginning, my disclaimer, that things are changing regularly. So notified bodies, when applying, they need also to keep on track on what is the current interpretation, the current state of the art. And since the publication of the regulation, even if it's not already, let's say, by date of application, because last year we got an additional year due to the COVID time. So the date of application was delayed to May this year instead of last year. We are still seeing a lot of guidance documents, and the regulation was 175 pages. But if you count the number of guidance that we received since then, it's actually much bigger than the regulation, and the new interpretation are coming. So the designation process of notified bodies is getting very tough to be able to get all the requirements. And every notified body who is applying need to show that they have competence in every single field. And nanomaterials is getting a special scope, which is a really specific code where notified bodies, when they apply, they need to show that they have competence in the assessment and the critical assessment of devices, which include nanomaterials. And this means that they need to show specific knowledge, and not just in the device, but also on how the device is manufactured, how it is processed, what are the technologies which are applied to be able to conduct such an assessment. So the qualification and competence level is increasing on the notified body side. Just as a short point, to give you also some ideas, because you are looking to innovation, you are looking for a future of nanotechnology, consider the fact that actually you need a notified body sector, not like going to EMA and you have a potential to speak in a pre-scientific advice meeting or a pre-meeting with the authorities. Notified are not allowed to do such things. And on top of this, during this COVID-19 time where travel restrictions are limited, it's not even allowed to do initial certification completely remotely, because the European legislator said, if you are doing this, it has to be case by case, and it should be shown that this product that you are getting an approval, let's say targeted for, should be actually a product which is necessary for the healthcare system and which can be justified that is actually needed and there is no alternative therapy. So you need to consider that timeline is also important when looking to new technologies getting to the market, at least at that time of COVID time. Later on, when travel are allowed, then it's not a big issue. But first of all, you need to find a notified body who's qualified for your device. Secondly, you need to find actually the right time to get an application for that notified body to get audited and get also the process initiated toward getting your device certified. Now, devices which are already on the market, which potentially include nanomaterials, they are not impacted critically because they still have a valid certificate. Nevertheless, there is a requirement that those devices need to be in compliance with the current state of the art. And this is where you could be challenged by the notified body and by the authorities when they look to your devices. I don't want to spend a lot of time on this. I want to step into the requirement on manufacturer and explain to you what you should be doing when you look to the medical device regulation. So every medical device manufacturer, regardless if they have, let's say, nanomaterial or not, they need to have a quality management system in place, which is according to the requirement of the regulation. Then they need to have technical documentation, which are in compliance with the requirement of the regulation. To be able to do so, they need to first of all understand, is it a medical device? Is it a medical device with a medicinal product? Is it a medical device which is actually with an intended medical purpose? Or is it a medical device which is without medical purpose, which means it's much more, let's say, like fillers, like liposuction devices? Those are also medical devices, but without intended medical purpose. After understanding that, you need to understand what class my device belongs to. And the medical device sector, you have devices which are falling into class 1, and you have devices which are falling into A, 2B, and class 3. And even in the class 1 classification, which is the lowest class, you may have devices with the involvement of notified body as well. And those are devices which are sterile, or devices which are reprocessable, or devices which are with measurement function. The class 1 solely without any of these functionality will be based on self-declaration. All others will be actually with the involvement of a notified body. So you need to understand the classification, because this would be impacting your device and the conformity assessment procedure which is applicable for that device. And then you need to understand what is the composition of my device, because it has an impact on your classification. Do you have nanomaterial inside your device? And nanomaterial is defined to be actually in the regulation to be a device which is containing particles which are between 1 and 100 nanometers. And it includes also a clarification that even other products may be also classified to be nanomaterial. So you need to consider whether you fulfill the definition of nanomaterial, and then you need to go to the further definitions, which might be also applicable for you, such as particles, which is defined in the regulation as well to be a piece of matter with defined physical boundaries. You need to look on whether you fulfill the definition of agglomerates, and then you need to understand, is it what do I have in my device is fulfilling that definition? And finally, you need to look to the definition of aggregates, because this is all of these definitions are connected to nanomaterials. And if you look to all of them and you recognize my device is actually including nanomaterials, then you need to look to the classification, and because the involvement and inclusion of nanomaterials will have an impact to the classification of your device. The regulation is saying whenever you have a device incorporating or consisting of nanomaterials, you need to look to the exposure of that nanomaterial to the body. So if you have non-invasive devices, none in contact with human tissue, then you have a negligible potential of internal exposure. As soon as you have invasive or implantable devices, and they have a surface area which can be deemed fulfilling the definition of nanomaterial, then you will move into the classification to higher levels. But as soon as you have nanomaterial in general, you're going to recognize when I have nanomaterial in my device, I'm directly under the assessment of a notified body. And if you remember from before what I said, involvement of a notified body means that you would need to have an organization who is first of all available to take your application and will be able to conduct an independent assessment in a timely manner, considering the fact that the timeline for the medical device regulation are very tough, and the number of notified bodies available as of today in comparison to the past is very limited. Now you found a notified body and you want to get your device approved. I don't want to deep dive into that. I just make it on a high level to make a clear understanding for everyone. Whenever you have a medical device and you want to get an approval, this means you need to have a quality management system. So you need to have the quality management system, which is enabling you to show that you fulfill the obligation which are defined in the regulation with regard to, let's say, compliance to management requirements, resources requirements, having established processes, documentation requirements, post-market surveillance requirements. All of these things you need to fulfill in your quality management system. You need to have also technical documentation which are enabling you to show that you have all evidence to support compliance of that device to the applicable requirements which are defined in general safety and performance requirements. The general safety and performance requirements in the medical device sector are nothing else than the minimum expectation that a device must meet to be able to get to the European market, to be able to be really placed on the European market as a compliant device. And for this, we have actually, like in the pharma sector, in the pharma you deal with let's say, monograph, ICH guidance, and so on. In the medical device sector, we have much more standards which are harmonized or non-harmonized. It doesn't matter. What matters is actually that the standards are reflecting the current knowledge and the current expectation, and manufacturer use standard to show compliance with the applicable requirements. So such as 10993, for example, for biocompatibility, 14971 for risk management. So this is how manufacturers show compliance with the applicable requirement. And the risk management standard is a key standard when showing compliance to the applicable requirement, because if you look to the regulation and you look to the first 9 general safety and performance requirements, all what you're going to recognize that everything is related to performance, safety, to a risk management approach, to a benefit risk profile. And this is important because it's key to justify that the device is safe and performing well when understanding actually the risk associated with that device, when understanding the intended purpose of that device, and what is the nanomaterial, for example, inclusion necessary for, and why the risk of inclusion of nanomaterial is still acceptable when looking to the benefit on a holistic level and looking to the benefit risk profile of that device. So risk management is key in the medical device sector. It's key also in the pharma. So it's everywhere key to have a risk management process. But if you look to medical device companies and you look to the medical device regulation, you see actually the focus and the necessity for establishing a risk management process, which is the quality management system, and ensuring interconnection between the different processes which are leading to a final conclusion on benefit risk profile of a device. There are specific general safety and performance requirements which are related to design and manufacturing. And this is where you need to pay a lot of attention when you are dealing with nanomaterials to what are the additional requirements that you need to meet by considering the material, by considering the chemical composition, by considering the biological, let's say, reactions that you could expect with such a nanomaterial if it's, for example, released from that device. And the regulation is including specific requirements, especially with regard to material which can be carcinogenic, mutagenic, or having an impact on reproduction. So they ask you to select your material very sensitively by looking actually on alternative materials to avoid any carcinogenic, mutagenic, or product which could impact reproduction and phthalate, for example. They tell you, it's not prohibiting you from using those kind of materials. It's telling you if you use such material, you need to have a very good justification. Why is it necessary? So you need to look to your nanomaterials, not just from a perspective that I have a particle or a material which is fulfilling the definition of a nanomaterial, but what kind of particle is that? What is the risk associated with the fact that that particle will be released in the human body? What will happen? What will be happening later on during the process? I can show you a lot of examples similar to what Renee was showing with regard to pharma. Those are the devices which were, let's say, coated, which were based on carbon and other materials, which leached out of the body and which led to actually a carcinogenic reaction. And this is where manufacturers need to understand the risk associated with every material, and especially with materials like nanomaterials, which can be released and absorbed by the human body and leading to further problems. And this is where manufacturers need to justify. It's not, there is no regulation and no requirement which is telling you you are not allowed to do something. The regulation is always telling you, think thoroughly about your process, take a risk management, let's say, decision mentality in every activity that you are doing, and try to address the risk accordingly in your holistic process. And the IFU and the labeling should be also ensuring transparency about what is included in that device. So if you have a device which includes carcinogenic, mutagenic, or product affecting reproduction, and they are over 0.1%, as it's mentioned here, weight by weight, then the regulation is telling you, you need to disclose that, even if you have a good justification for this. So you recognize that the medical device regulation is adding additional expectation on nanomaterials. It was already under the directive that there were some expectations, but they were not that much formalized like they are now with the medical device regulation, which is ensuring a higher level of expectation on devices, including nanomaterials, and ensuring that there is a higher oversight by notified bodies on such kind of devices. So all what you need to do when you have medical devices, including nanomaterials, understand your device, understand what kind of nanomaterials you have, understand the impact and the risk associated with that nanomaterial, and provide all evidence on verification and validation, which allow you to justify that your risk is on a level which is acceptable and you understand your device accordingly. And I think what is the most important part at the end is the validation via clinical evidence, because it's a requirement, and the clinical evidence is not just related to pre-market, but also to post-market strategy. I hope I could give you some helpful insight about the medical device regulation and impact on nanomaterial and product, including nanomaterial. Thank you.
Thank you a lot, Basil, for this very comprehensive overview on medical device regulations and the challenges that we had during the last years. I'm looking into the chat and I don't see any questions in the moment. So one question could be, you told us about the new medical device directive and all the challenges. So is there still a long queue of companies waiting for getting their old product to the market again?
Yeah, sure. I mean, you look to the numbers of notified bodies, and you remember 2012, like more than 80 notified bodies. Now we have nearly 20, and I see the question with regard to IVDs. We have actually just less than 5 notified bodies. The notified bodies increased their resources over the years, so they hired much more and more people, but it is still not sufficient because, as I mentioned, for example, for nanomaterials, you need to have specific expertise to be able to address that application. So the queue is long, And the notified bodies will be struggling in this short period of time to be able to cope with all the requests of manufacturers, especially with regard to completely new devices entering the market for the first time. So they are currently dealing with their current device manufacturers, and they want to get on top of new manufacturers, but they are human beings by the end, so they can't do more than at actually their capacity.
We have a question from Lorena. Lorena, if you want, switch on your microphone and ask it directly.
Sure, it was in line with this question that you just replied to. I was just wondering on for a young startup that is now struggling to get in contact with a notified body, should I start by getting the help of a consultancy firm? And also, I take the chance to ask a second question, which would be, how would nanomaterials actually influence an in vitro diagnostics product that is not in contact with the human body? Is this something that needs to be taken into account?
I mean, going back to what I mentioned with regard to risk management. So first of all, first question to your first question is, having a consultant involved depends actually on your capacity as an organization and your internal competence. If you have the internal competence and you have enough capacity, you don't need support. If you recognize based on your current capacity and the timeline that you have done for your project that you need external help, then select thoroughly a right consultant, because you need to find out a consultant with competence in this field who can support you towards getting your device on the market. To the next question, with regard to an impact of nanomaterials on medical devices or in vitro diagnostic, which are non-invasive, it's again a risk-based approach. You need to understand the impact of that nanomaterial on the product, on the performance of that product, and the safety of that product. And if you can show that this is actually not influenceable, like I said, you can have nanomaterials. You need to understand what is the risk associated with that, and risk-based, you need to come to a conclusion why it is acceptable. This is actually the whole mentality in the legislation in Europe. It's a risk-based legislation. They do not reject that you apply or use any materials. They tell you when you use them, you better have a justification why they are necessary. And if there is just a risk, then you will have a challenge because you will not be able to justify.
Thank you.
Sure.
Thank you very much. Thank you, Basil, for the presentation and for the discussion. And I think so we just saw Lorena, and so we go to the next presentation, which is Lorena's presentation. So I just want to introduce her. Lorena, she studied physics in Barcelona. After her studies, she went for a postdoc to Australia, and then she joined the International Iberian Nanotechnology Laboratory in Braga in Portugal, and she is group leader for medical device at this institute. And in 2018, she founded a company and she's CEO of those company, EURONANOMED. And this company deals with the early diagnosis and monitoring of cancer metastasis, and is one of, I think, one success story how young companies can succeed in getting nanomedicine to the market. And so I'm looking forward to learn more about this project and about EURONANOMED.
Thank you so much, Olaf. It is my pleasure to be here today to tell you about the journey that we have done at EURONANOMED from the, let's say, from the research institute into the company, hopefully soon into the market, actually. So I thought that would structure this presentation exactly as a journey. So I'd like to tell you about how the idea came up and where we were standing at that time and what is the current situation with the startup. So let me start by telling you about INL. So INL is the International Iberian Nanotechnology Laboratory. It's a research institute located in Braga in the north of Portugal that was established by the governments of Spain and Portugal back in 2005 to be the only international organization worldwide in the area of nanotechnology. We actually, in the health cluster at INL, we work in the interface between health and engineering. And what we do is to develop multidisciplinary technology to solve the society's challenges in the area of health. Our aim is actually to develop technologies that can help the clinical doctors either to diagnose or to treat a patient better as well as to help gathering further understanding about diseases. So in particular, in my research group, which is the group of medical devices, we work indeed at developing technologies for earlier and more accurate disease diagnosis, as well as for better understanding of diseases. And this is why we carry out these 4 independent research lines on the isolation of circulating biomarkers using microfluidics, on the development of nanobiosensors for multiplex analysis of these biomarkers, organ-on-a-chip systems to better understand diseases, in vitro, but in a mimetic environment, and then optical instrumentation to be able to obtain as much information as possible from these biomarkers, if I may say. Well, we have been working for the past 15 years in the area of cancer. There's no need to say that cancer is a major disease with a very high mortality and morbidity still nowadays, and actually this is mainly due to the fact that cancer, a primary tumor can be treated, but a metastatic cancer, or when cancer is spread to other parts of the body, the metastatic lesion is very hard to treat. There's one way in which cancer information can be retrieved at any time point for the clinicians to be able to tailor patients' treatment, as well as to provide a better diagnosis and prognosis of the disease, and this is through liquid biopsy. Liquid biopsy is actually the study of biomarkers of cancer that are obtained from body fluids. And an example is actually the study of circulating tumor cells, or CTCs, from the blood of metastatic cancer patients. These circulating tumor cells are actually shed from the primary or metastatic tumors, any tumor that is active in the patient. They travel through the bloodstream and potentially have the capacity of leave the circulation, enter another organ, and create metastases there. By studying these cancer biomarkers, for instance, the CTCs, one can get non-invasively continuous information about the tumor in a reliable way. And this is crucial, like I said, for the clinicians to be able to diagnose and treat the patients in the best way as possible. There's actually only one technology that is available in the clinic to provide liquid biopsy-based analysis of circulating tumor cells. And that technology is called Veridex by CellSearch. Actually, what this technology does is it isolates tumor cells that express EpCAM, epithelial cell adhesion molecule, by using magnetic beads that are immunoconjugated with this antibody, and it enumerates the cells. This technology is actually approved by the FDA to be used in metastatic breast, colorectal, and prostate cancers. And basically what it does is that if the number of circulating tumor cells on a given volume of blood sample is increasing, we know that the patient is not responding to treatment, while if the numeration of circulating tumor cells is reduced, then we know that the patient is now responding to treatment. And this is the only use of this technology. So again, it only provides enumeration. It can only count effectively epithelial cells that express this epithelial cell adhesion molecule, and the sample needs to be processed to be able to do this analysis and fixed. Hence, the CTCs are not viable afterwards. We decided back in 2004, when I was recruited to be a staff researcher at INL, in the framework of the Inventa project, that we could develop a technology based on microfluidics that would provide fast and gentle isolation of all circulating tumor cells from a whole blood with no pre-processing. Actually, the processing steps, being them centrifugation, red blood cell lysis, or anything of the sort, actually provides circulating tumor cell loss. Hence, we wanted to avoid all of that and process the whole sample, the whole blood sample, directly into the microfluidic system. We also wanted to capture all circulating tumor cells regardless of their phenotype, as it is well known that circulating tumor cells can run epithelial to mesenchymal transition, and hence not all circulating tumor cells are actually epithelial. And most importantly, we wanted to be able to run phenotypic analysis of the circulating tumor cells directly on the system, again, in order not to miss any circulating tumor cell. And we wanted to recover the cells intact and viable to be able to do functional studies. With this in mind, we created a microfluidic system that was based on filtration, taking into account that the circulating tumor cells tend to be larger in size than white blood cells and red blood cells, but also more rigid. So looking at the cell size and the permeability, we developed this filter-based microfluidic system that was capturing the cells that were larger or less deformable. In this video, you can see how the microfluidic system fits on a glass slide and is able to process a whole blood sample directly with no pre-processing. And on the video on the right, you can see how the cells that are labeled get trapped in these peelers in a very efficient manner. So we first, tested our technology with controls, as you do in the laboratory, using cancer cell lines, and we demonstrated that we could actually isolate just above 70% of the cells that we were spiking in a whole blood sample, while depleting above 99.99% of the white blood cells present in the sample, yielding a purity of about 10% that, despite it sounds low, was much better than competing technologies. At that point, we also wanted to compare our technology against the gold standard, which is the cell search that I introduced previously. So what you see in this slide is a comparison in a small cohort of metastatic colorectal cancer patients. So what you see here is that from each patient, we took 2 samples. One was analyzed with the cell search system, and the number of CTCs is presented in the yellow bars, while the other sample was analyzed with our technology, and the number of CTCs isolated you can see in the green bars. Needless to say, our technology was much more sensitive, but when actually plotting the survival of the patients against the number of CTCs that we isolated, we could see that cell search was not able to discriminate between patients, as they established a threshold of 3 circulating tumor cells per patient to be able to, let's say, diagnose the patients with a bad prognosis. But with our counting, let's say, we were able to actually separate patients into very clear patient populations, above or below 7 circulating tumor cells. And these 2 groups of patients had a very bad prognosis, but the patients with more than 7 CTCs actually had a much shorter survival, with a difference in more than 6 months. So with this, we decided that our idea was actually interesting, that it could potentially be certainly interesting for clinicians, and that's how we started participating in acceleration programs and in innovation contests. So we, to give you an overview, in a very short amount of time, we are talking back in the end of 2016, we participated in the NanoMed tab, nowadays called the Health Tech tab, promoted by the European Nanotechnology platform. And so in this case, this is a translational advisory board. We were looking at whether our technology would eventually have an impact in the clinic and whether it was worth to pursue. We, at the same time, participated in the Startup Nano acceleration program, which dealt with nanotechnologies effectively, and in the Resolve program that dealt with medical technologies. So we kind of had, let's say, a mixture of different profiles evaluating our technology from the nanotechnology point of view, from medical technologies point of view, and from both. And then we also were accelerated in the Startup Draga. So this happened in a very short amount of time between the end of 2016 and the beginning of 2017. And we learned some very crucial steps that we needed to take if we wanted to pursue this further. Those were to be able to protect our intellectual property. And for this, we submitted a patent application straight then to the European Patent Office at the end of 2016. We also needed to create a business model and a business plan to be able to see not only if our technology was worth for clinicians, but also a successful business potentially. We had to create a pitch deck. Of course, this is not a collection of documents that you create once and they are good to go, but they are iterative documents that keep evolving with the technology. And we also needed to produce a licensing agreement. Of course, a pitch deck is something very, very different of what you do when you're doing a scientific presentation. So it actually takes a new set of skills. And when it talks to a licensing agreement, of course, we realized that our technology was not property of the researchers, but it was actually property of the research organization for which we work. And hence, we needed to translate this technology from the research institute into the startup to be able to make a profitable business. While we were sorting all these 4 things, which I think are crucial for any scientist looking at entrepreneurship, we started also gathering further scientific evidence of the validity of our technology in the clinic. So it was also very crucial for us that this very small validation we had was extended to, larger numbers to be able to claim the scientific utility or the clinical utility of our technology, and also to convince investors to give us money to continue the development of this technology. So we did some tests also in metastatic bladder cancer, and we included healthy controls to show that the tumor cells that we were able to capture from all metastatic patients were never appeared in healthy controls. We also looked at what was the weakness of the other technologies, and we looked at how from the circulating tumor cells that we were isolating, only half or less were actually expressing ECOM, and this is due to this epithelial mesenchymal malfunction that I mentioned earlier. We were also looking at the molecular information that we could obtain from circulating tumor cells, and we realized, if you look at this graph on the right, that oftentimes, the most common situation is that the metastatic lesion cannot be biopsied. I mean, one cannot do a biopsy of the metastatic lesion because it's too invasive, because the patient is too poorly, or just because that tissue is not available. But what we see is that the circulating tumor cells are coming both from the primary tumor and from the metastatic lesion. So if we have the primary tumor available and active, the CTCs will carry molecular information the same way as the primary tumor. But if there's an active lesion, an active metastatic lesion, the CTCs will actually replicate the mutations, in this case, of the metastatic tumor. And this is very important because we know that in cancer, there's this clonal evolution. So if we want to treat a patient the best way, we need to know what are currently the molecular alterations of the tumor. And that's how we run another, let's say, preclinical test, this time in the metastatic breast cancer cohort with a much larger number of patients. And once again, we took two samples, one analyzed by cell search and the other by our technology. We confirmed once again that our technology was much more sensitive, but most importantly, we could also show the molecular expression of the circulating tumor cells that we had trapped and how this could be used as a companion diagnostics tool. So in companion diagnostics, what you do is to analyze the sample and recommend what is the best treatment for the patient in the light of this biomarker that you have just analyzed. So in this case, because this is breast cancer, we were interested in looking at HER2 status. So HER2 is a protein that can be expressed in some subtypes of metastatic breast cancer, and that it heavily alters the treatment that the patients are undergoing. I will not go in details, but basically what we did was to study the HER2 expression in the patients as they were responding to treatment or not responding to treatment. And so basically we demonstrated that the patients that were HER2 positive in the primary tumor, they could be positive in the CTCs or not. And according to that, they would respond to treatment or not. And the opposite, we could have patients that were negative, HER2 negative in the primary tumor, that would actually mutate after treatment, starting being positive and hence potentially benefiting from targeted therapy.
So we know we have some sound problems in the moment. So there's a kind of echo. In the beginning, it was okay.
Let me try to use some headphones to see if that helps. Give me a second. Is it working? Is it better now?
I think yes, yeah.
Okay, fantastic. Thank you. And sorry for the trouble.
Perfect. Yes, much better. Thank you.
Great. So basically, at that time, we had gathered further scientific evidence and we had developed our pitch deck. We had secured our intellectual property. We had secured also a licensing agreement, and we have developed a convincing business plan. We decided to incorporate the company. So EURONANOMED was incorporated in 2018. And since then, we have run preclinical trials in 8 hospitals in 3 different European countries, and we have run over 1,000 clinical tests, and we have also pre-orders. The roadmap for us now is to to scale up the fabrication of the technology in compliance with the regulatory framework. You have just heard about the IVDR regulation, so we need to fabricate our chip in compliance. And we are also developing an automated unit. We aim at finishing this development in 2021, so that then we can run registered clinical trials in 2022, and finally obtain the CE mark for in vitro diagnostics in 2023, and later on move away into companion diagnostics. So the solution now has to do with the Ruby chip. So this is the technology that we had at the beginning, was called the cross chip, and we have done several iterations on the technology to be able to be compliant with the CE mark. And we are also, together with the chip, working on the development of an automated unit so that the sample is processed and analyzed automatically, minimizing inter-user and inter-laboratory variability. Most importantly, these technologies together would be able to provide from the sample to result a report to the oncologist in less than 3 hours with still a very high efficiency and with cost efficiency as well. So ever since we founded the company, we have won several innovation awards, and we have also gathered funding from competitive funding programs like the European Commission. We are currently running 2 European projects. We also got support from the La Caixa Impulse Consolidate program, and then we also have seed funding from venture capital, as well as the several innovation awards here and there. We are currently developing our automated units with the help of the Caixa Impulse program. So, like I said, we have a technology that is fully automated and does not rely on an operator. And we are also working on the evolution of the chip. I just showed you that we are working currently with this 4th generation of the technology. We went through several iterations, and we are currently actually scaling up the technology with the help of another European project that will actually change the material with which we are working, ensure the quality control of the technologies, follow the ISO standards, and also decrease the price. Still, we are still looking for funding before we are able to bring the technology to the market. We need to certify the technology in the light of registered clinical trials. And for that, we need to finish the mass fabrication. So the prototype that we were using was fabricated in-house, but the one that we need to use for the IVD certification, as I said, in compliance, needs to be done elsewhere. And hence, this is crucial for us. And of course, the funding for this is not negligible. Just to finish, I would like to introduce you to the team. This is, of course, not a woman show in this case. So there are 3 people that were involved from the beginning in the creation of EURONANOMED, and those are the 3 founders, Sara Balde, Paulina Piairo, and myself, that we gather all the technical competence to develop the technology, but then to bring the technology further and to be able to actually have a successful product, we needed to incorporate profiles in the area of business and also in the area of quality control and regulation. And that's how Enrique and Liliana joined the team. So far, we keep running trials in different hospitals and in different research projects as well with universities and other RTOs. But I think the most important thing about our technology is that the oncologists that have been working with us are very, very interested in our technology. They keep asking us for more samples to be processed because they have noticed that with our technology, they can stratify their patients and they can actually personalize their therapy so that they can give them the best opportunity as possible. And that's why we keep working on this. And that's all I had to say for today. Thank you very much.
Thank you so much for your presentation and for this really interesting and successful project. Very impressive.
I'm so sorry for the sound issues.
No problems. That's the web conferences. They are like that, but they became much better in the last month. In the beginning, it was much more difficult.
Yeah.
So I'm looking for questions, but I don't see any questions in the moment. So do you think the hardest part of the work had been done, or is the hardest work still to come, because now the amounts of money becoming bigger that you need to go ahead for the manufacturing and things like that?
I guess, you know, the hard job is always the one that is still coming, because the other one has already been achieved. Now, I think we went through many loopholes, and it took a while to re-educate us as researchers to be entrepreneurs. That's probably something that could be done better. Generally, in Europe, if you compare us with the mindset in the States, it's really different. I think in the States, they normally keep in mind their, you know, potential entrepreneurship from very early on. For us, we really needed to be re-educated to be able to learn all these things. Otherwise, we could have gone much faster to the same maturity state that we have now. I think, of course, the steps to come are very difficult. However, I believe that we have partnered, because that's the thing. One has to recognize their strengths and their weaknesses. And by recognizing our weaknesses, we have been able to look for partners that can help us to fulfill those needs. And I'm talking about, you know, having excellent advisors from the oncology side, as well from the pharma side, since we want to pursue the companion diagnostics strategy, also from regulatory point of view, from many different you know, complementary fields, also from the corporates, from the diagnostic and the pharma industry. So hopefully we are able to gather this economic support that we need to be able to fulfill our roadmap. And I would be pleased to tell you a few years down the track that that our technology is actually placed in hospitals. Hopefully that's the case.
Thank you. So I see there's another question in the chat. The question is, you showed a patent that was filed in 2016. Isn't this patent getting too old for a good protection?
It's a fair question. We are actually, we have a second patent that was filed in 2017, and we are working towards, again, towards bringing that patent from the research institute into the startup. So most of the new patents that we file are always from the research institutes, and so we always have to go through that process. But also we are working towards having patents that are sole property of the startup as well. And we hope that those will see the light now in 2021. But indeed, one has to build not just the one patent, but a portfolio of patents that secure not only the technology, but also the freedom to operate of the prospective product. And we are working in that line.
Thank you. So I'm looking again. I don't see any more questions. So thank you again for this really nice talk.
Thank you.
And the next topic is the coffee break. So I'm sorry that due to the coronavirus, we only can offer you the break, but not the coffee. I hope that you have the possibility to get some coffee where you are. And then we start again at 4:30, so at 4:30, with the next talk about Live Nano. So please enjoy your coffee break and see you again at 4:30.